Um, Thank you, Lou, for inviting me, wherever you have lost track of me, Lou. And thank you, Jackson, for the introduction. Further to that introduction, I'm here tonight, it's totally accidental. In 1976, January 16th, I was an unemployed archaeologist. And I got a job working in the Department of Botany at the Royal Museum for Dr. Candries. And I had a job typing. And he had $400 in his petty cash, and he paid me $100 a week for typing for him. Well, I found out I was typing the bibliography of a PhD student who was just finishing up his dissertation. And I had it done in 3 days, and he thought the money was for 4 weeks. And so he hummed and hawed, and he was puffing on his pipe. And he said, just more typing over there or something like that. And the following day, he found some money through the Department of Archaeology, through the museum, and hired me part-time to work as a lab assistant. And about 2 days later, myself and another archaeologist who was doing pollen analysis from sites in Ontario, he was from Manitoba at the time, and I actually knew him. He said, I want you and Don to set up an archaeobotanical identification course on Saturdays for the Ontario Archaeological Society. And it'll last 10 weeks, and you'll be introducing students from the Ontario Archaeological Society and how to identify charred seeds and nutshell and wood. And I was saying inside, what is archaeobotany? What's the other archaeobotany? So, interestingly, I had no biology background. I was an unemployed archaeologist. I finished my undergraduate years at UT, and I started looking at seeds from, actually, interestingly, the name of the archaeology site was the Seed Site in Woodbridge area. Anyway, I started identifying the seeds and identified some raspberry seeds and some elderberry seeds and I think an acorn nutshell fragment that I was hoping to move. And that was my journey. It just started accidentally, and I was there, and I was just, Jock was bringing in this material from archaeological sites, and he was saying, I can set up an identification, what's the word I'm looking for? What's the word I'm looking for, Margaret Ann? Service, thank you. This is my wife, Margaret. She knows more than I do. Anyway, we set up the service, free identification service, and John sent 10 items. And the archaeologists from Ohio and New York State and Ontario were sending 10 bags of samples. So I just plowed through it, unknowingly, what am I doing here? And I just thoroughly loved it. And I just kept on going through it. And all of a sudden, I had a body of data. And the doc was saying, you have enough information here to maybe, I say, Rudy, try your master's degree using this information. It's archaeology and it's botany. And in that short time, I applied at York University and I was accepted. And I used the information that I had gathered, something like 25 or 30 sites in a very short time in the early 70s, late 70s. And in 1981, I started my master's degree at York University. But since then, I've been doing it on my own for the archaeology community all over Canada. You just keep on sending the material, and I identify it, and I send reports, and keep on sending the material. So this is all well and good, but I wasn't earning a living doing this, and I had to get a job. And I went back to the university to get a master's to get employed in archaeology. That didn't happen. And being unemployed again, I was hanging around Margaret Ann school, doing all sorts of work for the teachers, for the principal. The principal asked me to do education, archaeological and archaeobotanical education courses for the board, so I did that for the elementary school panels and high school panels. I was just doing all these things, and all of a sudden I was in the realm of elementary school, and people were saying, well, once you go back to school and get your teaching degree, so you can earn a living. Anyway, age 50, a young woman from the Ontario Institute of Child Studies at UT, and she took me by the hand, and they introduced me to the folks at ICS, and there was 400 applicants, and 100 were chosen, and then 40 of us were interviewed, and I was one of the 40. So I got, it was a 2-year program at the time. Now it's a 2-year MA program. And I finished, did really well, and I got a job. Within 3 years, I was in supply, but I was, again, another learning experience. I was doing all the things I do as an educator. I think I've been an educator my whole life, and when I look back at it, all the different jobs I had had. Anyway, I just kept on doing this, and got my teaching degree. Then I taught supply for 3 years, and then by 55, I full-time employment, and my wife's quite a school for a short time, and then I got my own position and retired 10 years ago. But all the time I was doing botanical studies on my own. And since I retired, he actually paid me, because when I started doing this 4 years ago, there were no archaeology companies in Ontario. Now there are about 100. And they hired full-time archaeologists and part-time archaeologists to do some field work. And I'm one of the few specialists to do this. And so a lot of different companies asked me for my services, and they paid me well. And I've been lucky enough to get so much information. That's why I looked at material from all over Canada, and it's as I speak, I have 6 contracts that I'm working on right now, and that's going to be taking me into the new year. And my wife's leaving at the end of the time, but I'm trying to relax myself and try to figure out where I'm going to go with this. But I will show you some slides on this. I don't want to go too much. What I do, here's something. This is these. What you're going to be seeing here are slides, not a PDF program, because you can see the type is over other type, and then PDF, I've been told it just locks it in. So there may be a few samples, a few slides that may be overlapped. We'll just see. So what I do is I identify carbonized plant remains. So during their stay in the soil, they've been burned, and they've had contact with fire, either in the soil or actually been thrown into the fire. You may be all familiar with traditional archaeological artifacts, ceramics, projectile points, animal bone artifacts, and animal bone, and these all allow us to anchor a particular cultural group in time and space. Settlement patterns also another major dimension of archaeological research, to find out where the people lived, how many people lived in an area, how large was the village, and so on. Too heavy on. I'm jumping around, I don't know why. I'm trying to. This is a. You might have seen yourself driving by, somebody shaking. What is collected here, you can see there's some charred wood remains. You may find the odd hickory nut that's been missed during hand excavation and so on. So we do collect a little information from that. Most of the material is collected by water separation. So here's me. This is 1978 at a site in Stouffville. I think I collected material from 1,000 samples from longhouses mostly, and palisades and hearths inside the longhouses. So that has to be water separated. So this, what happens is a lot of the material just sticks to the surface tension, and we collect that, dry it out, and then we place it under the microscope and start identifying the plants. Sometimes there's a lot of heavy material, charred wood, for example, and other biological remains. But for the most part, we don't have time to do that, but this is just laden with most of the material that the archaeologist is looking for. And then here's an idea. All this material here is collected and dry it, and then labeled, of course, because we can't move the ingredients because it's not very meaningful if we don't have any location. 78, and I'm just getting used to this. There's a double bucket. Here's a laundry tub, and then there's another second laundry tub, and there's window screening in the bottom, so you can throw the soil in and then swish it around like you're panning for gold, and then the seeds and what have you float to the surface. This is a more sophisticated one here today, but a lot of companies now are building their own. And so the material is, there's the soil being thrown in, and the seeds are all, usually this water is turbulent, so it helps move the seeds and nutshells and so on in there, and it's collected down in there. I'll be really good at this. Here's my microscope. These little flags here, I have 24 boxes, and they have all the tree species. So when I'm identifying a fragment of wood, maple goes in the maple box, beech, and so on. And then I count and weigh everything, and then I graph the information. You see some of the work at the back, and I'll show you some more examples here as I go through these. So it takes, we have to collect the material, and then when I go into the lab, sometimes I have to sieve it again through a series of smaller sieves to, it facilitates identifying the material because if it's all the same size, it's very easy to pick out the raspberry seeds, and the tobacco seeds, and peach seeds, not peach seeds, but that's historic, wild plum, and so on. And so this kind of gives you an idea of what I find in my petri dish, shell, and this is a raspberry, it's a modern counterpart. This is how I identify all the material. I have a reference collection in my head, but I also come across seeds all the time that I'm not familiar with, so I have to identify them as well. So I need an extensive reference collection of modern seeds, and here's one that I was using at the museum down in Northern Ontario, and then my own wood collection where I've actually had the wood charred and made samples for identification. This is really useful when I'm training students to do this. So I use modern reference material and also the modern reference literature. And these are the cultivated plants that I find from the archaeological record, and above the photographs you can see the relatively early time periods that they've arrived in Ontario from their origins in southeastern Mexico. One of the important parts of identification is identifying the various elements of some plants. For example, I can identify corn by 12 or up to 16 different elements I can identify corn from. But that's very small fragments. So the fragmentary evidence is as important or more important sometimes, because if we're looking at time periods where there's the early introduction of plants, sometimes you don't get entire specimens. And if you can identify a variety of elements of a particular plant, then that allows you to make an identification, tentative at first probably, but then if you have more and more information, then you build up your confidence and then you can confirm the identification. Beans from a site in Coote County. We identify squash seeds, but also the woody stems from the squash. These are particularly prevalent, these squash stalks, in Paytoon Wyandotte sites. And when I'm looking at the material, I'm constantly finding stems as well as a few seeds. The seeds are rarer than the stems. The seeds are very fleshy, as you know, and they have to be carbonized, and the burning has to be stopped before they're entirely consumed for us to find them. So they're very rare. Sunflower seeds. The occurrence, some numbers, like if you're going through, for example, if you're going through a sample, you may 20 is a large number on a on an archaeological bed of sunflowers. 2 or 3 or 4 squash seeds, or a large number. Corn, you may have dozens or hundreds, and then you may have thousands of fragments as well. Tobacco are really small, and this is a modern tobacco seed with all its sinuous lines, or as I've been told by grade 1 students, I can see the wiggly lines there. So I use that in my talks sometimes. You'll notice the wiggly lines on the surface. And here's the archaeological specimen of these wiggly lines. They were first identified by a high school teacher in Toronto, again working in Dr. Cameron's lab at the Botany Department of the ROM in 1975. So relatively young-ish, because I know when I was first starting, archaeologists weren't finding tobacco hardly at all. It didn't come in until years later when we started using flotation, where it's found on many sites. Well, I haven't identified wild rice yet, carbonized wild rice. But Dr. Crawford at UT, who teaches Palaeoethnobotany, and has since 1987, I believe. And he has identified wild rice from a site. Interestingly, it's the seed site, the one I was working on in the mid-70s. And just 2/3 of a wild rice seed. It's definitely a very long, you notice that wild rice is a very long grass seed. We find the native plants are really interesting. Large fleshy fruits, small fleshy fruits, and then we get seeds of also a variety of weedy species that are, to some are weeds, but they're actually food. Just about everything was used by First Nation groups. It still is. These are some of the more obvious seeds of wild plum from 2 sites in Simcoe County. This is a Peytoon Wendat site, and this is a Huron Wendat site. Hawthorn, we find it not in large numbers, but it's a very obvious one because it's interesting. Once you get familiar with looking at the material, you'll see them right away when you're looking through your petri dish. The large seeds stand out. Then there's a mid-sized seed, and oh yeah, we're here. And it's the small ones that give you some problems because you actually have to use your microscope. And I've been very, very lucky with the one I've had. I've had it for probably 35 years. And I've been very envious of some of the microscopes I've had. Fortunate enough to look at different universities when I visit archaeology and anthropology labs. Here's some of the other plants that I identified. Wild grape, we don't have large numbers. Wild raspberry, we do. Strawberry, I'll have large numbers, but not as many large numbers as wild raspberry. Dogwood is rare. Sumac has a respectable showing on many sites. Spikenard it occurs sporadically and usually in small numbers. And elderberry, same. Elderberry and strawberry kind of tied for third. Excuse me, in numbers when I'm looking at the material. My samples that I get from the archaeology community can be 5 to 400. And you can see that the ones that have 400 take a long time to go through, especially if they're more than one item. Here's some of the smaller. characters in the assemblages that I look at. Blueberries are not very common. Brambles, I told you again, there's a nice close-up of strawberry with these finger-like lines on the surface that is not identified surveyed. And this is a nice photo of an elderberry seed that Brienne Rifle was actually mentoring her this past winter. She's a grad paleobotany student who's now employed as a paleobotanist for an archaeology company in Ontario. And here's some of the wild plants that are used for pot herbs, or just they can eat as greens off the plant, or they can cook with them. Nightshade, parts of it are edible, but can also be used for medicine because it is toxic. There's some of the weedy species you might be familiar with. I don't know. Purslane, if anybody knew had purslane, it's in the Spanish family, and I was told, go ahead, try it. I was reluctant to the end. Anyway, I enjoyed it. And there's a close-up of a nightshade seed and wood sorrel. These are very uncommon. I've only identified maybe a handful in 4 liters. These are relatively common, especially in the early sites. When you go back 4,000 years and coming forward in time, 4,000 years ago to 2,000 years ago, most of the plants that were prominent in the archaeological assemblages are nuts and berries. And all these have been identified from the archaeological record, as you can see here. And here's some carbonized material from sites I've been working on. From the site in Kent County, I did this when I was working at the Museum of Military Archaeology in the late 70s, and identified these hickory nut shells from a site in northwestern Ohio. Also, when I was working at the museum for Dr. Finlayson at the time. Acorn. You can see it very, very small. He's cranked up the image just so you can see these little lines here, these striations, and you can see it's coming apart from the actual outside shell casing. And then the nut shells, especially acorn and hazelnut, are not very prominent. They are there, but if one is going to find them, they're going to find them in the very old sites. Those sites 2,000 years ago and back into time to about 5,000 years. I also started examining the cross sections of trees, not knowing anything about trees, and I started a lot of these slides that I've been putting together for the last 2 or 3 decades are eventually going to end up in a book. And I've been creating various and sundry book quasi-books for students so they can take the information with them to do the work they want to do for an archaeological company, for example. And I can do it very quickly. I can create all these things very quickly. And in fact, when I had students over this past winter, I was doing them on the fly. They were doing the microscope work, and I was creating reference literature for them to take away with them. So I identify the different kinds of woods. The deciduous woods, we have the diffuse forest woods where the pores are spread throughout an annual ring. And here's the ring forest woods. This is the early wood, or the spring wood of red oak. And then this is the summer wood here in between. And then this is the spring wood, summer wood, and so on. And when you're looking at deciduous woods, they tell you right away who they belong to, what class, where you're going to place them, what box you can put them in. And then we have the conifer woods. Most of what I find on the archaeological assemblies is pine wood. And most of you probably know pine was a good fire starter. And it wasn't used for structures so much as other conifer woods like cedar. But we often find it in hearths, and we find it in other features inside of long houses. And back in the mid-60s, mid-70s, I created this little identification kit. These are modern American specimens that were actually carbonized and labeled. And then you can take your archaeological specimen and quickly go up and down to identify, to see, compare the cell structure of your specimen, the archaeological specimen, and the reference material. And the next several slides just show you a variety of diffuse and rainforest wood, sugar maple, beech. I see these aesthetically as well, that sometime when it be doing some large paintings using the cross sections of these lace-like structures. Elmwood is particularly a favorite of mine because it has these wavy lines. They actually go like this. You can imagine a large tree up and down and up and down, and that ulmiform, that wavy line, is anchored in the four elm species that we have here in Ontario. Red oak. In my career so far, I've never identified, I didn't touch anything. I haven't identified a pine seed. I've identified a pine wood, but I don't have any pine seeds, but it'll show up. This is the earliest period we have, where we have a lot of information from plant information from this time period. And these are the sites that I worked on, with the exception of this one here. These wooden stakes on the bottom are part of the fish weir, and they were put at the Atherley Narrows. I think I have a picture of it coming up to collect fish, and that's the wood is dated to 4,600 years before present. And carbonized material that I looked at was from this site over near Ottawa, the Muldoon site, which is around 5,000 years before present. And then I've had a smattering spread through southern Ontario. This is actually adjacent to Rice Lake, and then there's a whole series of them coming down into southwestern Ontario. This is a particularly interesting site, the Davison site. I identified a single black walnut fragment in 1976. And there's an anthropology professor, archaeologist, Chris Ellis, who's just about ready to retire, has been excavating this site in the last 5 or 6 years, and he may have the largest late archaic site in eastern North America. He has biological material and archaeological artifacts that are going to tens of thousands, maybe into the hundreds of thousands. But the biological remains are really fascinating because he has such a the site was interesting because they had these subterranean 30 or 40 foot diameter buildings. So all that subterranean scars in the earth filled up with biological material, and that's never happened. Usually, late archaic sites are samples from a random fire, a campsite. And so this is going to be probably the late archaic site in eastern North America for the next generation. These are some of the plants that I've had the pleasure of identifying over the years. A number of fleshy berries and 5 or 6 different kinds of nuts and a lot of different kinds of woods. When identifying the wood remains, it also tells us, it paints a picture of the environment on a particular site at a particular time. So if you had working with an artist, you could actually look at it looking at the artifacts and material and then having direct evidence of the plant and animal remains are really interesting, paint a really interesting picture of location in time and space. So some of the graphics I've been using and experimenting with for looking at and displaying the information in some interesting way. Actually, putting the pictures on this for an audience as opposed to putting it in a report. I guess I could put it in a report. I haven't decided. This is from the Muldoon site, the very old one. There's nut remains, mostly butternut shell remains. You can see that I identified almost 2,500 fragments of butternut and only 150 nutshell fragments and just a few hickory nutshell fragments. But this is a large body of data from a later archaic site. And then it's the tree species. And this is identified, obviously, mostly sugar maple, a smatterings of birch and ash, red oak, some pine. There's a larger image of the wooden stakes from the fish weirs at Jackaning, Chickening fish weirs. I think. Don't quote me. Here is the everything arrows here. So at this little juncture, you have these system of wooden stakes that you could actually prevent the fish so that they could go in and collect them. And here's up close and personal pictures that were taken by Jim Conley, who's head of the Department of Anthropology Department at Trinity University, and Kate Doherty is the lab tech there. And in the area, in the Hamilton, Burlington, Brantford area, I've done some work over the years and collected information for tonight's presentation from other archaeologists and other paleobotanists. This is, I identified a single nutshell fragment from this mid-archaic site about 4,000 years before present, and just 4 different kinds of wood. So it wasn't very much information. This is done for an archaeology company in Ancaster. This is the archaeologists call this the Middle Woodland period, and about 2,000 years before present, and identified a few bramble seeds and a couple of goosefoot or chenopod seeds, I think a single stargrass seed, and a variety of tree species. From a feature like this is an example of a feature from the site. And here are some of the tree species in detail from Miller's Pond. Again, you can see that sugar maple is kind of dominant in the oaks, red oak and white oak, with smatters of beech birch, and poplar, willow. Sometimes it's very difficult to identify some trees from one another with the fragments you have, because they're very, very similar. We have only a certain area on the surface of the cross section to work with. And in this area, or Botanical Gardens area, there's 3 sites that I'll be talking about. The Bulls Point site, which was excavated by Dr. David Smith at U of T in 95-96. And the information I'll show you is from the 96 period. And then the nursery site, which is currently being, well, in the last several years, Dr. Scott Martin from McMaster University has been teaching the field course at the nursery site. And what we hear is particularly interesting because it's the closest point site. And up until 10 or 15 years ago, it was the earliest site where we had evidence of corn. And but since we've been using flotation and the archaeologists have been deliberately doing research looking for early corn in Ontario on the Grand River, especially, and in the Hamilton area, we have placed those dates from the 6th or 7th century back into maybe as early as the 3rd century. And here's a nursery site from this feature, the Scott Martin and several students, these are the plants they identified. They had a number of fleshy fruits, and then some greens, amaranthus, and goosefoot, St. John's wort, which is a medicinal plant, purslane, wood sorrel, yellow foxtail, and a few nut remains. And this is from Larry Gaspardi. She was a student of Scott's several years ago. 2009, wow, time flies. Must be getting old. The Princess Point site, David Sutter was a graduate student at McMaster. He was the first, him and the kid, Bill Fox's friend, name escapes me anyway. Grad students, they were in Princess Point in '69 and found some evidence of corn, a number of corn kernels. They had some other plant remains. And they sent that information away to paleobotanists at the University of Michigan. So back in the '60s, most of the Ontario archaeologists were sending material to the University of Michigan and also into Illinois to have identifications done. There's nobody here doing that at the time. And not until myself in the mid-'70s and another as a high school teacher as well. And then Dr. Colbert started the Palaeoethnobotany program at U of T about 1986 or 87, I believe. And then across the pond, so to speak, at Coote Point, these are the plants that were identified. Amazing. They weren't sure whether they had squash or not. And a number of fleshy fruits, and some greens and grains. They didn't do the wood charcoal that I'm aware of. And then Redeemer College, I did this several years ago. There's an archaeology company in Lancaster which asked me to do the work. And so it's just some of the information I gathered from the report. And gave it to them and also gave a copy to Redeemer College, to the biology department. These are actual photos of the material from the Redeemer site. This is the tree species identified. There's 300, over 300. And you can see sugar maple and beech are prominent. And lesser so, ironwood and some birch. With this information gathered from a number of archaeological sites in the area, one can draw conclusions about what kind of canopy lived under. Maple and beech seem to be the dominant species that we find when we have a body of data from several sites. So they know that the area was maple beech 1,000 years ago, 2,000 years ago, and so on. I've been doing in the last, off and on, up until recently, I was doing 19th century Euro-Canadian sites, but now it's part and parcel of what I do. Equal amount of First Nation sites and also Euro-Canadian sites in the last several years. This was done in 1985. They actually wrote it up in the Flamborough News at the time, I think. This is the trees I identified from the Foundry area. So ash, elm, maple beech, and willow poplin, some birch, and some ironwood. Dropping over to say hello to the Brantford folks. This is the same Lancaster company I'm working for today as I speak. This is very precipitous, this hill. I had trouble walking up it, so one of the stronger workers had to have his hands strategically placed on my hindquarters so it wouldn't fall backwards. Anyway, I made it, and it's a struggle because I don't have knees like I used to anymore. So anyway, it was my site visitation, and I'm currently this material, and hoping to have it done by Christmas. It's a Christmas present. These are some of the plants I identified from the site. I don't know if you can read them or not, but there's a lot of, these are the charred wood species, sugar maple, and the local characters that I usually identified. Maize and tobacco were found. I only found 3 tobacco seeds. Wasn't a lot of maize, but it was there. And then these are some of the charred seed remains and the berries. It was interesting. The hill was kind of like that, but it went down like this. And I had to identify some of the river levels over the last 3,000 or 4,000 years. And the more level components of the hillside showed that they were there for some time. You can see there's a lot of material built up. This is all pottery remains. This is that mystery object. This is totally accidental. I normally just look at light fraction material, as I explained, and then sometimes heavy fraction. And I was privy to some shaker material. And this was stuck in a piece of mud. And it was just sticking out of the heart of it. Very fluid. And so I got a teasing needle and an artist's paintbrush. And I excavated it at the micro level and saw this part here. I was saying, ooh, do I have a bead? And when I got it excavated, it revealed itself. That falls right through and it exits there and you can see through it. But this is the oldest artifact I've identified in 43 years. And I think it might be the oldest wooden bead in eastern North America. I'm just saying. I have to find that. I say it with confidence now because nobody's told me any difference, but I want to do some research and ask some folks from south of the border if I'm allowed to. Sorry. And this is this is all in the recent past. In fact, the back table there's an article that I was telling an archaeologist, and he explained to me, oh, I'm the editor of the Bead Forum, which I hadn't heard of. And he was my boss in 1974. I said, well, write me a little article on those two things, because I'm showing these cool pictures. So he wrote that in. It's an international Bead Forum on my newsletter. So it's been, now you're famous all over the world. I said, maybe more infamous than famous. Anyway, you can see it's beveled, and on both sides it's beveled, so it was manufactured as opposed to have been eaten by some sort of rodent like a squirrel or a rat or a mouse or something like that. And I've shown it to numbers of archaeologists, and that particular whole style exists on other artifacts like shell beads and animal beads. The same structure. It looks identical. That's one way I was very convinced that it was an artifact, not just a wild plant seed. In addition to the seed. This was a piece of stitch bark from the beaten site. Actually, I was working on that site in 1976. And this is from the Draper site. I've identified 3 of these in 40 years. And they have this perforation. They're about this big, maybe that big. And they're very obvious and they're carbonized. Of course, I can't break it to identify the wood. I poke around the edges with a paintbrush, see if I can pick up some pore structure. Sometimes I can make a vague identification. This is diffuse forest wood, but that's all. And these are some of the sites I'm looking at. Bill Finlayson asked me to look at the material. This book is at the back as well, and there's something like 800,000 or 900,000 seeds. And boy, it takes a long time to count them by ones. I'll probably come up with another better way to count them at some time. But it's mostly berry seeds. Like these are, here I say, I've been privy to a lot of privy material. There I said it. Sorry. But it's mostly berries and jams and fruit that they're using in the household and dumping into the curtain. And this is the, here's some data. Almost 700,000 raspberry seeds, and you can see they're all in the thousands here, and other berries, less and so on. And this is the rotation device that they used at the time. So down in there is the soil, and she's stirring it about, and all the seeds come here and they collect it there. This is from a parking lot site from downtown Kingston. Identified a whole raft of plants. This is an interesting site because I had my first Afro-Canadian seeds. It was the first time I had a coffee bean from an archaeological site, but I knew it instantly because we had some stored just recently. Anyway, these are some of the fruits and cultigens, squash seeds, sunflower and coffee beans, and so on. And an offshoot of this, over the years, people started asking me, can you come up and talk to my class? So that was elementary school, and then high schools, and then universities, and then grad programs. And I even had the pleasure of talking to, in 1980, grad students and staff of the faculty at the Botany Department at Western. One of the PhD students wrote, can you come and talk about archaeobotany? We don't know anything about it. And I was talking to them about seeds and carbonizing. They're looking at me like this. They didn't see botany like that. They were the scientists looking at the material and the structures at the micro level and so on, even further down than I was. So Margaret Ann and I have been all over Ontario working with First Nation groups and schools and ladies' church groups, Flower Society, that's Flower Society. Redeemer College, Dr. Chang asked me to come and talk to her students as well. So actually, I can't believe I'm looking up there and seeing 2013. Just see the this is Six Nation group. This is what they have now. Now they have a larger quarters to where they have guest lecturers. And Margaret Ann and I work with the archaeology community who work training First Nation students and monitor liaison jobs on archaeological sites. So they're the go-to people when they have something of note that may remotely look like human remains, and they make the decisions right away. Stop digging. And many archaeologists that I know, when they see something like that, they know right away. They'll stop it themselves, and if they can make the identification. But usually they get identification first when they're touching it, and then it's either buried or they make arrangements with the First Nation group that's closest. If they're going to be moving the remains, they have to bring in the coroner because it may be a recent burial. They have to have the police involved, and the chief, and often it's a combination of everybody working together. And that's the milieu I live in now, is working with First Nation groups and having a close contact with them and sharing my knowledge for a long time. This is at the Kids Day of the ROM with the Ontario Archaeological Society. So we have kids old and young. The youngest I've had is a 2-year-old. I looked through a microscope. And the oldest was 92. This was a 2-day stay at the Science Museum in Ottawa. I was asked to come down there and set up a mini lab. If I may just, as an aside, I'll be setting up a mini lab in mid-October, the 19th and 20th, out in front probably, here. Dr. Gilbert is setting up a 2-day archaeology conference for the public. And I'll be giving a paper, probably something similar to this, but also I'll be setting up 2 days of mini lab outside, so people can look through a microscope and see my knowledge. For several years, I was asked to come down and put an annual forest festival And that's one of the early sites where I had actually some wood and seeds for them to look at. These particular ages are really interesting to me because I'm so used to looking at material 20,000 years old. When I started looking at material 2,000 years old, 3, 4, because we don't have much information still from those older time periods. This was, we were asked to come down to a science program set up for inner city First Nation kids on a Saturday, and this is on campus at the University of Windsor. And I had a lecture downstairs in their dungeon, and then we all went over to the biology lab, and the director of biology greeted all the children and said, "I want to see you here in 10 years." And then the director of education came down, and he was looking at story. I have 17 storyboards, and I use them as instruction. So that I had material from an archaeological site, which is really cool. And the 15 students had it all laid out on the tables, and they can go to the pictures and try and identify a maple seed, a corn seed, a bean seed, and so on. And that was particularly gratifying. The education director was most impressed. Here we are working away. We even had a grandmom. She brought her nephew over there, or grandson. And so I had her slaving away, separating charred remains, which was kind of cool. And this lady here was one of the instructors. She helped us. And this is Russell Nadee. He's the director of Aboriginal Studies at Windsor, and he asked me to come and participate in their science program. Dundas. I was recently there just this past year. Yeah, there again. And then when I was asked by Dr. Gupta, she sent me an email, and I saw Lakehead. Lakehead's about a 10-day journey from Margaret Ann, and I figured I can't do that. But they had a campus, a satellite campus in Aurelia, so I'll skip it and jump in mid-January. We're very lucky. Or mid-March. So I brought everything. I had all my storyboards, and I had material from an archaeological site. So I had them actually scrutinizing everything and identifying seeds using my reference collection. And I have a lot of constantly collecting nut remains and seeds and plants and putting them in bags and labeling them. And they can be laid out and they can be used for comparative purposes. And I had Dr. Gupta looking at some tobacco seeds. This was you can see it's jumbling there. That has to do with being a slide as opposed to a PDF slide. Anyway, I was looking at this material with Dr. Abel and identified tobacco, and this was really interesting. Tobacco doesn't occur in large numbers. It occurred, I had 208 tobacco seeds from just a very few samples. He was over the moon about having that much tobacco because also on the site were hundreds of tobacco pipes. And we found out later that this is a particularly interesting location for this site because all the various groups coming through the area had to come here to get permission to move through their territory. So they were sharing smoking pipes with one another. And we discovered this tobacco seed, which was really cool. And that's one of Tim sent that to me. So there's a tobacco pipe and there's a tobacco seed from the site. This was taken by Dr. Captain Patterson here at McMaster in Sustainable Archaeology. So I have a really interesting, nice working relationship with all the various groups here in Hamilton. They've been very grateful for me to be. I have 25 minutes, you ready? That's it. This was also at the same presentation, where we had all sorts of children and adults looking through the microscope. Dr. Gary talked about Ward and Bonnie Cross. In 1990, Gary was my boss and at the Ministry of Transportation, and Bonnie was a field worker and I was working in the lab. So we're all kind of stoked. And just 2.5 years ago, we had 18 Mississauga elders come to our house and I gave them a slide presentation, and Margaret Ann made chili and cornbread. Wonderful. They were there for about 3 or 4 hours. We shared stories, actually. Some of them are quite elderly. I find myself being elderly, but they were elderly. And again, this is Bonnie. Here's Bonnie to the cross now. I gave a presentation in her class just here at McMaster. Shanty, Morel Hart just moved in from California 2 years ago, and she appointed me to the visiting scholar status in the department because I'm giving lectures there all the time and working with students. And then 2 years ago, at the end of the year, Phil Fox, the professor at Trent, invited me down. I thought I'd be giving a 2-hour lecture and then go for lunch and so on. Head back to Toronto. But he had me working from 9 o'clock until 5 o'clock. He would take us over there and we stayed overnight. And then last year, we're invited up to the Nipissing First Nation for their Aboriginal Day. And I set up shop at the back, and that's me waving my arms about and so on. And this is really nice because it was just last fall. And they had an archaeology day, and I had a huge room, so I had lots of room. This is a dad showing his son how to identify wood. And I'm using the wood bender that I have outside there. And these are the 3 students I've been mentoring over the past winter. They've all gone on. Brianne is now working as a paleobotanist at an archaeology company in London. Tim is my child company. And this young gentleman just came back a couple of weeks ago, a month ago, from Georgia, Georgia near Russia, not the one near Alabama. And he was there for a 5-week archaeology course. He's a gifted student. I can see him going right into the PhD program as soon as he's finished his undergrad. And last but not least, Laura, she's also interested in ethnobotancy. She's doing her graduate